Discuss about a topic that is histochemical techniques. So before knowing about this histochemical techniques, we need to understand what is histochemistry because these histochemical techniques comes under this branch of science histochemistry. So histochemistry is the branch of science concerned with the identification and distribution of the chemical constituents of tissues by means of stains, indicator and microscopy. So it's the branch of science concerned with the study of chemical composition of animals and plant tissues, particularly the distribution and the location of chemicals within and between the cells by means of stains, indicator and microscopy. So it is an important technique that is used for visualization of biological structures, and it makes the use of chemical reactions between laboratory chemical reagents and cellular chemical constituents such as carbohydrates, proteins, lipids and nucleic acids. Some of the goals of histochemistry are it is used for the presentation of normal chemical composition and distribution. It determines the specificity of reaction products and also helps to detect the reaction product too. The techniques and methods used in the histochemistry. So, histochemical techniques, we know that there are different techniques used to stain the cells or tissues to observe the color structure under the microscope. But however, these cells can't be directly stained. So before proceeding with the staining part, there are some steps involved in coloring a cell. They are first one, animal treatment and tissue processing, and second one, fixation of tissues and cell. Third one, the process of embedding and sectioning. And the last one is staining and observation of specimen by microscope. So these are the steps involved in coloring a cell. So histochemical methods are based on the type of molecule needed to be studied on the structure to be observed. So in this topic, we mainly going to discuss about the three important histochemical techniques. They are the demonstration of carbohydrates, the demonstration of proteins, and the demonstration of lipids. Demonstration of carbohydrates. For this, we use periodic acid stiff test. So periodic acid stiff is a staining method that are commonly used in histopathology laboratory. So it is used to highlight the molecules with high percentage of carbohydrate content such as glycogen, fungi, basement membranes in skin, like that. The purpose of this test to stain the glycogen present in liver, kidney, skeletal and cardiac muscles, like that. So the principle of this test is that the glycol group of carbohydrates are oxidized by periodic acid to release dialdehydes. So these aldehydes on subsequent combination with Stiff's reagent results in the formation of a magenta colored complex or the color ranging from red to violet. So this intensity of color demonstrates the tissue component with carbohydrate attachments. So this is about their principle. And the reagents required for this test are periodic acid and Stiff's reagent. For the preparation of periodic acid, first of all dissolve 0.5 gram of periodic acid in 35 ml of absolute ethyl alcohol and add 0.2 sodium acetate and 10 ml of distilled water. Keep the solution in the darkness at 22 degrees Celsius and use it at a room temperature. If brown appears, discard the solution. So this is about the preparation of periodic acid. For the preparation of stiff reagent, we need fetsin basic 1 gram, distilled water 100 ml, and 2 gram of sodium metabisulfate, and 2 ml of concentrated HCl, and at last 0.3 gram of charcoal activated. First of all, dissolve this basic fetsin in boiling water and cool at 50 degrees Celsius and filter it. After that, add sodium metabisulfate and con HCl. Store at dark room temperature overnight. Add charcoal and shake for 1 minute and filter it. Then the reagent is colorless and it stains free aldehyde from red to violet. The color of the reagent gradually becomes pink on exposure and its staining properties will be impaired. So these are the reagents used for this test. So next about the procedure. So let us see how this process will be take place. Flame the slide on the burner and place in the xylene. Repeat this treatment to remove the wax. After that, drain the xylene and hydrate the tissue section by passing through the decreasing concentration of alcohol baths and also in the water. After that, place the sections in periodic acid solution for 5 to 10 minutes. Rinse these sections in the water and treat with stiff reagent and cover with stiff reagent for 20 to 30 minutes. And again rinse this in the running tap water for 5 to 10 minutes. After that, cover this with hematoxylene for 3 to 5 minutes. And dehydrate after that dehydrate these sections in increasing concentration of alcohol and put these slides into xylene baths for clearing. At last, mount this in BPX or other any mounting medium and observe under the microscope. So at last we can see the shades of purple or red to violet color could be observed in the tissues, and this shows the presence of carbohydrates. So you can see this figure, it's a stained tissue that's showing the presence of glycogen, that's the carbohydrate. So this you can see a deep magenta or shades of purple or red color or violet color, and this is showing the result. This is showing the result of carbohydrates. So this is the formation of insoluble magenta colored complex that denotes this positive result. So this is about the demonstration for carbohydrates. So next is the demonstration of proteins. For this we use mercuric bromophenol blue method. This bromophenol blue indicator, it's a phenolphthalein anionic dye that binds to protein under neutral to acidic conditions. So this bromophenol blue method determines the protein level in samples, especially in solubilized cultured cells and cell membrane proteins. First, so let us see the principle. This bromophenol blue is an acid dye and can normally combine with the basic groups of proteins. And the mercuric ions of this bromophenol blue solution react with acidic and aromatic residues of this protein to give blue color. So this is about the principle. So next we can see what are the reagents used for this method. First one, mercuric bromophenol blue. So this mercuric bromophenol blue is made by dissolving 1 gram of mercury chloride and 0.5 gram of bromophenol blue in 100 ml 2% aqueous acetic acid And the fixative used for this test is Carnot's fluid or 10% of formalin. Next, about the procedure. Bring these sections to water and stain in the mercury bromophenol blue for 2 hours at room temperature. After that, rinse these sections for 5 minutes in 0.5% of acetic acid. Transfer the sections directly into the tertiary butyl alcohol and clear in the xylene and mount in TPX. You can see the result of a deep blue color in that sections. It shows the presence of proteins. So here you can see the presence of protein. This blue stained tissue showing the presence of proteins, and these four figures are the kidney. These four figures are the kidney sections that are stained with mercury bromophenol blue to demonstrate the proteins. And you can see this bottle. This is the bromophenol blue indicator. So this is about the demonstration of proteins. And the next one is demonstration of lipids. For that we use Sudan black method. And this black Sudan is used for staining of wide variety of lipids such as phospholipids, sterols and neutral triglycerides. And this black Sudan can also used for chromosome staining, Golgi apparatus and leukocyte granules like that. Let us see their principle. Sudan black method is widely used for the demonstration of lipids, and Sudan black B is a fat soluble diazole dye, and that can be used for staining of neutral triglycerides and lipids. They are slightly basic because of its amino group, and it combines with the acidic group of compound lipids such as phospholipids. So the coloration of lipids by any oil soluble dye depends upon the reagent dissolved in lipids. And the reagents, so this is about the principle, and the reagents used for this method is Sudan black B and alcohol. Prepare this saturated solution of Sudan black B in 70% of ethanol and keep aside for a week. And also we use 70% of alcohol. See how this test will be take place. Stain the sections in Sudan black B for 15 minutes. Differentiate in 70% of alcohol until a delipidized control sections appears colorless. Dry it and mount in the glycerine jelly. Treat the pyridines extracted materials in the same manner. On the slide in glycerine and observe under the microscope. So you can see a black or dark blue colored stained tissue showing the presence of lipids. So on microscopic examination, varying degree of black colored pigments are seen in the positive reaction. So this is the method, that is the Sudan black B method is used to demonstrate the presence or absence of lipids and fats. So these are all about the different techniques for the demonstration of proteins, carbohydrates and lipids. Thank you.